We will be looking at the toxicology of codeine. Codeine is an analgesic alkaloid that is used in the treatment of mild to moderate pain. It has both natural and synthetic sources, as it could be extracted from the latex of the opium poppy, or could be synthesized by methylation of morphine. Codeine has many medical uses. It is found in common cough medicines, painkillers, and is sometimes used as an antidiarrheal. The most common route of entry is the oral route, but parenteral administration is possible. As codeine is administered primarily through the oral route, its absorption occurs mostly through the GI tract. Based on its structure and properties, we can look at what might affect its bioavailability. If we look at the functional groups, codeine has two ether groups here and here. These have polarity rankings of 8, so they're relatively unpolar. Codeine also has an alcohol group here and an amine group here. These have polarity rankings of 3 and 6 respectively. Overall, these functional groups give codeine nonpolar characteristics. In terms of its ionization, codeine has two groups with the potential to be protonated. The amine has a predicted pK of 9.19, which is higher than any pH in the GI tract, so it would most likely be protonated. Similarly, for the alcohol group, with a predicted pK of 13.78, it would most likely be protonated as well. Thus, codeine would mostly be an organic cation through the GI tract. Finally, if we consider the log POW of codeine, which is 1.2, it also has hydrophobic tendencies. So as a whole, codeine is a lipophilic organic cation. And while that may point towards transcellular absorption via the organic cation transporters, recent studies suggest simple diffusion of the neutral form of codeine or transport mediated by cation proton antiporters are more likely the methods of codeine absorption in the intestinal epithelium. From the intestine, codeine travels to the liver via the hepatic portal vein. It retains the same lipophilic cationic properties and crosses most membranes through simple diffusion. It is finally absorbed by the hepatocytes, and the liver is the major site of metabolism for codeine. So let's take a closer look at that. In hepatocytes, codeine can be metabolized in a few different ways. Most of codeine, around 80%, is metabolized through conjugation by glucuronidation reactions. It is catalyzed by a glycosyltransferase, UGT, at the phenol group, and this makes codeine more polar. Another 10% of codeine is N-demethylated to norcodeine, and this is catalyzed by the enzyme cytochrome P450-3A4. A final 5% of codeine is biotransformed to morphine. This involves O-demethylation catalyzed by cytochrome P450-2D6. These final two reactions are oxidations, which expose the alcohol and amine functional groups respectively. But morphine can be then further metabolized to normorphine through N-demethylation by cytochrome P450-3A4, or to major metabolites, the glucuronidation products morphine 6-glucuronide and morphine 3-glucuronide. These reactions are again catalyzed by UGT. So if we look back at this diagram, after metabolism occurs, some of the metabolites will be eliminated. Through metabolism, the compounds have become more polar and water-soluble, especially the codeine and morphine conjugates. As such, they can no longer pass through the membranes by simple diffusion. So to be distributed towards excretion pathways, the metabolites are transported out of the hepatocytes by the multi-drug resistance associated proteins. The compounds are circulated to the kidney where they will be excreted in the urine. Studies indicate that 95% of a single dose of codeine will be excreted within 48 hours, predominantly in the form of codeine 6-glucuronide, unmetabolized codeine, and morphine 3-glucuronide. The codeine metabolites that aren't excreted may go on to exert their activity. The target of codeine's toxicity is the mu-opioid receptor. However, codeine shows a very low affinity for this receptor, so it is through the metabolites that it exerts its mechanism of action. This makes codeine a prodrug and the parent toxin. Until quite recently, codeine's effect was believed to be caused solely due to the conversion to morphine. Now, morphine 6-glucuronide and codeine 6-glucuronide are also considered active metabolites. Due to the target's primary location in the central nervous system, the codeine metabolites would be required to cross the blood-brain barrier in order to reach it. While the transport systems have yet to be identified, passages of these metabolites across the blood-brain barrier may be mediated by organic cation transporters or proton-cation antiporters. There are 4 different types of opioid receptors, but the specific target of codeine is the mu-opioid receptor, which is a G protein coupled receptor. These are primarily distributed in the central nervous system, the spinal cord, and may also be expressed in the GI tract. These receptors are involved in pain-modulating pathways, as their activation leads to the inhibition of neurons and pain transmission. They are activated by endogenous opioid peptides such as endorphins, which are released by the pituitary gland. Endorphins and their activation of opioid channels cause the so-called runner's high. In normal physiology, an opioid receptor is activated by the binding of endogenous opioid peptide, causing the associated G protein subunits to dissociate from the receptor and act on intercellular pathways. The alpha subunit interacts directly with a gated potassium channel, opening it, increasing the potassium flux, and hyperpolarizing the cell. This would inhibit further action potentials as there would be a greater stimulus required to surpass the threshold. Another subunit of G protein dissociates. This is the beta subunit, and it binds to the calcium channels. It reduces the pore opening of the channels, inhibiting calcium influx. Additionally, the G protein inhibits adenylyl cyclase activity, which decreases cyclic AMP levels and further reduces calcium influx. With reduced calcium influx, the neurotransmitter release will be inhibited at the synapses, so signals will not be able to pass across the synaptic cleft. Through the modulation of calcium and potassium flux, the signal transduction pathways of the opioid receptors affect neuronal excitability and transmission. In the cells, hyperpolarization inhibits the action potentials, and at the synapses, reduced calcium inhibits the release of neurotransmitters. So overall, the binding of opioids inhibits stimuli transmission across neurons. Morphine and the other metabolites of codeine are opioids themselves, so their mechanism of action is the same as endogenous opioid peptides. They interact with the mu opioid receptor and activate the inhibitory actions of the associated G protein. So when morphine 6-glucuronide, or codeine 6-glucuronide are in the vicinity of one of these opioid receptors, impulses through these neurons will be inhibited. Depending on where the interaction takes place, the different areas of the brain, the spinal cord, or even the intestines, there can be different effects at the organism level. The primary effect observed is pain relief, and accordingly, one of the major uses of codeine is as an analgesic to treat minor to moderate pain. It provides this relief because the activation of the mu opioid receptor and its associated inhibitory G protein prevent the transmission of pain stimuli. So if we look at the pain stimulus pathway, the mu opioid receptors in the spinal cord may be activated by one of the codeine metabolites. This could prevent neurotransmitter release at the synapse and inhibition of further transmission of the stimuli. Opioid receptors in different parts of the brain also play a role. They inhibit the perception of pain and further spinal cord transmission. When administered as an analgesic, there are often unwanted side effects such as euphoria, caused by the inhibition of GABA release. GABA is an inhibitory neurotransmitter. Without its release, there is sustained excitement of selected neurons. This type of side effect could lead to addiction and abuse of codeine. Codeine and other analgesic opioids also have the potential side effect of constipation, and for this reason are sometimes used as antidiarrheals. The cause of constipation with the treatment of codeine is the mu opioid receptors located in the GI tract. When these are activated in the intestine, the peristaltic action is inhibited and slows down the movement through the bowels. Another common use of codeine is in cough syrups. Coughing is suppressed by codeine because its metabolites interact with the mu opioid receptors in the respiratory center of the brain. When sensory neurons send stimuli to the central nervous system because of irritation, opioids inhibit the transmission of the signal to the respiratory center. As the signal never reaches the cough generator, no stimuli is transmitted back down, and no cough will be initiated. These are the major therapeutic effects of codeine, but if the recommended doses are exceeded, the same mechanisms can lead to toxic effects. Use of codeine and other opioid drugs can lead to addiction and abuse because of the euphoric side effects they have. In these scenarios, overdose is more common because tolerance develops. When an overdose of codeine occurs, the same effects are observed but to a greater degree. The most common cause of death due to codeine toxicity is respiratory depression. This is again caused by inhibition in the respiratory center of the brain. The excess codeine metabolites would cause sustained inhibition of these neurons, eventually leading to respiratory arrest and death. If you are looking for more information, these are the articles where the information was gathered from and readings that can be done on codeine and opioid receptors.